We're working in two different areas. We're working to edit genes inside the body with in vivo delivery and to create edited cells and use where the product is the cell itself as a therapy. So in vivo, we are working on a kind of amyloidosis called transthyretin amyloidosis. Now, amyloidosis is the kind of disease where a protein folds in an unusual way and often creates, they self-aggregate and form clumps that precipitate. And depending on the protein, they'll precipitate in different parts of the body, but it's the precipitates themselves and where they deposit that drives the disease. In the case of transthyretin amyloidosis, these proteins will either accumulate in peripheral nerves or primarily in cardiac tissue. And so you often get this peripheral neuropathy phenotype or this cardiomyopathy phenotype, but sometimes you get a little bit of both. So we know from clinical studies that the role of this protein, the normal function of this protein, is fairly minor in the human body. And so a strategy where you just get rid of it entirely is a viable strategy. And so that's what we're trying to do. We're trying to knock out the production of this protein, this mutant protein, because in people who have hereditary forms, they carry a mutation that predisposes to this aggregation. And so the strategy is to just knock out the protein and suspend the further production and therefore the further creation of these deposits. So that's our lead program on the in vivo side.
May I ask, how close are you to applying for clinical trials on that?
So we're hoping to file our IND in the second half of 2019, so next year. So we are, I think, making very nice progress in building all the tools necessary to bring that forward. The approach we've taken in terms of the delivery has used lipid nanoparticles as opposed to viral-based delivery. And so that means there are more elements that we need to sort out in the progression of that form of delivery to the clinic. We believe, though, that will give us a kind of modularity as we go forward beyond that first program, and that we'll be able to flip second and third programs out of that very quickly. So we think that extra upfront time is going to be worth it downstream when we get into other conditions. On the in vivo side, we've been working with our partners, Novartis, to generate therapies in the hemoglobinopathies space. So that includes sickle cell disease and beta thalassemia. And that's where we're editing hematopoietic stem cells. Primarily the thought now is in autologous transplant. We are also working with Novartis on making better versions of CAR-T, a very popular engineered cell type right now, which is where there have been 2 approvals for CAR-Ts for immuno-oncology applications. We redirect the, if you will, the targeting of the T cell to a particular tumor. And there's always been thought that you could do more with the cell when you have it in hand than just retarget it. So you want to boost its performance, and that's what we're helping Novartis do, to create essentially second-gen versions of CAR-T products.
You're also looking at a form of leukemia and tumors, cancerous tumors.
That's right. So recently this year we announced a progress in our work in our wholly owned program on the ex vivo side, where we're developing a T cell that, rather than using a CAR to redirect it, we're using a recombinant T cell receptor, TCR, which is the natural way a T cell is targeted. So the strategy is to use a TCR of our choosing to direct the T cell to a different kind of tumor, and this is called AML. It's the most prevalent kind of tumor in adults, that occurs in adults. And the interesting thing that people have noted with CAR-T therapies is more limited, perhaps, success in the solid tumor setting. And a number of people think that maybe the TCR-T is the better strategy to pursue solid tumors. And so we want to leverage this primary reactivity towards that we're creating. So we're targeting a particular antigen epitope that's going to be found in the AML with the TCR, and that is an epitope that's also found upregulated in many other kinds of solid tumor. And so we hope to be able to take this initial work, and as we successfully bring that forward for AML, to be able to leverage that and bring the same general reactivity, but maybe tuned to different environments as we think about different kinds of solid tumors, but with the same targeting.
And what does the timeline look like on that?
We haven't reported on that timeline. Since we're trying to do more than just create a recombinant TCR in a regular T cell, that takes a little bit more work. We reported on the initial success in finding the right kind of TCR that looks good, and the initial in vitro and a characterization of those cells. And so we're building on that. Obviously, we want to bring that forward into the clinic as quickly as we can, and we're working very hard to do that. But we haven't guided on when that would be in the clinic yet.
Hepatitis B is another area you're working in?
That's right. So as we think about expanding our success in getting into the liver, so maybe just to take a backward step, the big challenge with CRISPR is, particularly when doing in vivo delivery, is achieving successful assembly of the editing machinery in a given cell type. That's a very large hurdle. So we've solved that, I think, by and large, for liver delivery. So essentially, we're in the liver. Now that we're in the liver, what else can we do when we're there? So there's a variety of other diseases that we can pursue with a knockout approach that are genetic in nature. And we reported recently on some progress, at least in mice, in terms of knocking down those proteins. And these would be alpha-1 antitrypsin deficiency and in primary hyperoxaluria type 1. But we also are interested in HBV, which isn't a genetic disease, except it's an infectious disease. And one of the challenges with HBV is the reservoir of the viral genome that sits around in people even when the active replication of the virus is being adequately suppressed. So you have to stay on meds essentially all the time, because if you came off, it could reestablish itself from this reservoir. And so there's long been this thought that perhaps what we really need to really sort of sterilize the liver, quote unquote, is something that can eradicate the viral genome that's left inside hepatocytes. And CRISPR has been thought to be a great tool for doing that. And so we've been exploring that as well.